So next we're going to hear from Jan Friedman, who's going to talk about FORGE, or Finding a Rare Diseases. What is the last thing?
I'll show you.
The E stand for, I should know this, right? This is a project actually that we developed in partnership with Genome and CIHR, and we were so thrilled with how this has advanced and turned out, and actually has really put Canada on the map as leading internationally in this area, and so we look forward to your presentation.
Okay, so the E is genes. We cheated. So this project, which we are very proud of, and I'm pleased to have a chance to talk to you about, is now just 21 months old. It began in response to a joint call by Genome Canada and CIHR called Advancing Technology Innovation Through Discovery. This was focused on childhood diseases for which the genes could be identified quickly in a small number of subjects. This was an attempt by both CIHR and Genome Canada to demonstrate application quickly. The applicants had to be pan-Canadian disease consortia and had to include at least 2 of the Genome Canada Science and Technology Innovation Centers. There were 2 projects that were funded, one in rare pediatric diseases that I'm going to tell you about. That's the FORGE Canada project, and the other in rare pediatric cancers. The FORGE project began in a workshop in June of 2010 that was called with about 2 to 3 weeks notice. We were able to get representation at that workshop because there was money available from every single clinical genetics unit in Canada. We had 80 physicians, still have 80 physicians and 50 scientists from all over the country involved. The consortium that was put together, which is called the FORGE Canada Consortium, is led by Ken Boycott at the University of Ottawa, Jacques Michaud at Saint Justine, and I are the co-leads, and there's a steering committee that's shown here with representatives from Calgary, Toronto, Newfoundland, Montreal, more Montreal, and more Toronto. So people from all over the country, including not only clinical geneticists, but also bioinformaticians, genomicists, ethicists from throughout Canada. So this really is a national consortium. The purpose of the project was to use next-generation sequencing to identify genes that cause rare diseases in Canadian children. Along the way, we hoped to improve the bioinformatic pipelines, the approach to analysis, and also there was an ethics research component that was embedded within the project. The project was originally funded for 18 months. It was extended now to 24 months. Excuse me, began the 1st of April 2011. So it's now just over 21 months old. A key component and a unique component of the FORGE Canada Consortium is the clinicians. As I said, there is at least one clinical geneticist at every clinical center in Canada, and in terms of organization, one clinical geneticist was asked to lead from each center and served as the primary contact for that center. Each center recruited patients or families for sequencing and also recruited subjects for validation cohorts, and I'll return to that point in a moment. These were recruited not just through the clinical genetics services, but rather through any of the clinical services. It's a pediatric project, so largely pediatric services, But the intention from the beginning was not to restrict this to diseases that may be seen in genetics, but throughout the pediatric hospitals. And many of the conditions that we have sequenced were brought forward by other kinds of pediatric clinicians. The key role of clinicians in this project took advantage of previous national research and professional collaborations. There are only a little over 100 clinical geneticists in Canada, so the people know each other professionally because patients tend to move from place to place, and we talk to each other at professional meetings. But more than that, there have been a number of research collaborations over the years that involve all of these groups, funded by CFI, funded by CIHR, and these have gone on for over 25 years. There have been these national collaborations of various kinds among clinical geneticists, so that it was possible for us to get all these people together in the same room within 2 weeks, and to get them on the same page and for the same project after a 2-day meeting. The other reason that the clinicians from all over the country were important is it allowed us to establish local expertise in gene identification and also take advantage of local expertise that exists both in gene identification and functional analysis to try to understand what these genes do. So the idea is that the clinicians know who in their academic community is working with fruit flies or C. elegans or yeast or mice or other model organisms to understand these diseases. The embedded ethics research component, which Martha Knopers leads, produced national guidelines for patient-family consent for research studies involving whole exome or whole genome sequencing in children. And the fact that it's in children does add another wrinkle that Martha brings a great deal of expertise to. And also fostered a common approach to sharing these results with the families. So the way we provide the results back to the families. And from the beginning, the intention always was, and has been, is, that although this is a research study, the reason the families participate is not out of the goodness of their heart necessarily, but they want to find an answer. And so we feel an obligation to give them an answer, even if it's we didn't find anything, or we found things but we're not sure what they mean. So in every case, the obligation that we have to the families that participate is to feed back to them whatever we found, to the extent that we can. The project is involves 3 categories of gene identification. The first group are unrelated patients with the same disorders. So there are many rare genetic diseases that are highly characteristic. These are things that even a clinical geneticist sees once or twice in her career, that any genetics center may have a handful of patients, no more. But it's not possible to get enough patients to study without a consortium, a national consortium. So these are groups of patients who have very rare diseases, but very characteristic diseases that we have some reason to think have a Mendelian etiology. The second group are multiplex families that are classic genetic kinds of families where there's autosomal recessive, X-linked recessive, or autosomal dominant inheritance, consanguinity if they're recessive inheritance, and we're able to use the tools of linkage to narrow down the region where the candidate genes may be. And there are a number of families we studied like this. These are families that actually have been accumulated at various genetic centers for many years, and we hadn't been able to go further than linkage studies on them. And the third group are autosomal recessive disorders in genetic isolates. More so in central Canada and eastern Canada than out here, there are genetic isolates, groups of people. Some of them are native groups, some of them are religious isolates who tend to marry within their own groups. So there's not close consanguinity, but there is a limited gene pool, and there are characteristic autosomal recessive diseases that occur in these groups, and the likelihood is that although the families think they're unrelated or appear to be unrelated, the same founder effect, the same gene is involved. So in these cases we can sort of combine the previous two and use the tools looking for linked gene or areas of homozygosity, and also different families that all fit together in the same way. Let me just give you a couple of examples from BC and how this worked. One is Weaver syndrome, which is one of these rare, but very characteristic conditions associated with intellectual disability, generalized overgrowth, marked enlargement of the head, characteristic facial features. About 40 cases have been reported in the 35 years since this was described, so it's very rare. It's usually sporadic, but there's one report of a parent-to-child transmission which suggests that it could be a dominant disease. These are what these kids look like. These are two children that were sequenced in this particular study, and the one on the left is actually the first patient who was ever described in Seattle 35 years ago. Bill Gibson did this work in Vancouver. He's one of the clinical geneticists here. He, through the FORGE consortium, he sequenced the exomes of two trios, child and both parents, necessary to sequence the trio because we were looking for new mutations, and we wanted to find a mutation that was present in the child and not present in either parent. There were 24 or 25,000 different genetic variants found in the exomes of each of these two children. 17 of these in one child and 16 in the other had never been seen before and were de novo. They weren't present in either parent. But there was only one gene where there was a de novo variant that was present in both children. And that was EZH2, which happens to be a gene that's involved in cancer that the Genome Sciences Center here has considerable experience with, and started jumping up and down when they saw this gene come up. They subsequently tested a few more patients and found another de novo variant of the same gene. So it clearly is the gene that's responsible, at least for a large fraction of patients with this particular condition. This paper with Bill as senior author and the FORGE consortium was reported last year in the American Journal of Human Genetics. The second example are sort of a set of related syndromes we call Megalencephaly Syndromes. These are syndromes in which the brain is enlarged. The children are also enlarged. They're overgrowth syndromes. But the brain in particular is very much enlarged. There often are abnormalities of the cerebral cortex. Vascular malformations of the skin may or may not be present. Abnormalities of the limbs may or may not be present. We've looked so far at two particular subgroups, one called MCAP, which means Megalencephaly capillary malformations. So they have little vascular skin lesions, usually not terribly noticeable, but invariably there. And the other is called MPPH, which stands for Megalencephaly polymicrogyria, polydactyly, and hydrocephalus. So they have abnormalities of the cortical architecture of the brain, extra fingers, extra toes, and also hydrocephalus in addition to the large head. Studying this group of patients in a number of different individuals, and this was a study that Lindley Armstrong, one of the clinical geneticists here in Vancouver, contributed to, and Margot Van Allen, another of the clinical geneticists, suggested actually to FORGE. Three genes were implicated. PIK3CA, which was found in 24 of 29 patients with the capillary hemangiomas. 15 different mutations were found, and every one of them was mosaic. So this is a condition that we think is lethal if all the cells in the body are involved. But if only a subset of cells in the body are involved, then you get this condition. Mutations, one particular mutation, it's actually a substitution of glycine for an arginine, or an arginine for glycine, at position 373 of the amino acid of the PIK3R2 protein. Same mutation, new mutation, found recurrently in 11 children with the Megalencephaly polydactyly and polymicrogyrias syndrome. And also 2 children with the same clinically identical syndrome were found to have mosaic mutations of another gene, AKT3. Again, only mosaic. It's very interesting because these 3 genes are all part of the same pathway and very closely related to each other. And we learned, we had no idea that any of these genes were involved in development of normal brain structure, and now we know that they're all 3 critical, and other candidates in this pathway become candidates for causing disease. And moreover, this is a pathway, a signaling pathway, that there already exist molecules to modulate, and it opens up the possibility of potential therapy. So, this is how we found these. These are examples of things that we found. Once we found them, the question of how you prove it's the mutation that actually causes the gene, I told you there were multiple examples even of de novo mutations in all of us. First of all, the changes are all confirmed by Sanger sequencing, an orthogonal method to prove that we don't have some kind of technical error. We use genetic approaches by comparing other members of the family and making sure the mutation is either de novo or inherited the way we expect it to be. We do genotype-phenotype correlation, where we look at children who have a particular phenotype, look for other children who have the same phenotype, and see if they have the same genetic change. Both the phenotype and the genetic change are extremely rare. And if we find them together repeatedly, we've got the proof. And the other approach is functional, looking at animal models of normal or abnormal gene function. It turns out that the genotype-phenotype correlation has been absolutely critical, partly because we've been in a big hurry. We've been trying to get these answers out quickly, and we haven't really had time to develop animal models from scratch. We've used some in some cases where they already exist. But the other reason is that animal models don't always recapitulate the story in humans. They tell you a lot about the pathways and the mechanisms involved, but they're not always faithful replicates, frequently are not faithful replicates of exactly what you see in humans, because mice are not humans, and fruit flies certainly are not humans, and yeast are even less human. So we do the best we can. And it turns out in this regard that the National Consortium has been very valuable in terms of getting additional patients with the same condition. And we've also established or taken advantage of a number of international collaborators for these confirmatory studies. So both in Canada and in Europe and the United States, there have been many opportunities where people have collected patients of a particular kind. Once we find a candidate gene, we've been able to talk to those other groups, collaborate with them, test additional patients, find additional cases with the same mutations, and prove that we're dealing with the right thing. So here's a status report. As of the end of Q7, December of last year, sequencing has now been completed on about 200 different rare pediatric conditions. Testing has been performed on a total of about 600 families, either whole exome sequencing or in some cases locus-specific validation studies. Diagnosis has been provided in about 300 of these families. So we have information that we can return to the families that participated in these studies in about 300 instances. We found 80 disease genes, found and validated 80 disease genes in this 21-month period. 22 of these were novel disease genes never seen before. 26 of these are known disease genes that were associated with either a novel or unexpected phenotype. We've encountered again and again genes that we know that cause a particular disease, but now they're causing a different disease too. And sometimes it's a radically different disease. There's probably other factors involved. We don't know what those are. But it turns out that many of the genes, as we know, have multiple different effects on the body, operating at different time in development. So this really doesn't surprise us, but we keep running into it. We've also found 32 known disease genes with known phenotypes. So some of the families, particularly in the consanguineous families or the genetic isolates that we've studied, they have a condition that is not so rare, but can be caused by many, many, many different genetic mutations. So for example, deafness. There are hundreds of different genes that when mutated can cause deafness. And exome sequencing provides a way to find which one is involved in a particular family without having to test those 100 genes one at a time. And in a genetic isolate in particular, in that group, once you find out what gene is involved, then you can test other members of the community and find it very, very inexpensively. So it's useful doing this for that reason too. In 120 conditions, they're still under analysis. 22 of these have novel genes that we think are causative, but we're still trying to confirm that. In 82 cases, we've got the opposite problem. We've got multiple variants, and we can't figure out at this point, haven't figured out yet, which particular one of those multiple variants is in fact causal. And these are cases where we need more collaboration, we need more cases to test, and sometimes we need animal models to sort it out. There have been only 16 instances where we didn't find any plausible candidates within an exome. These may be instances that are very genetically heterogeneous. They may be instances where there's not a genetic cause, or the genetic cause is not all that's involved, or the genetic cause lies outside the exome. We've published 14 papers so far in the 21 months, 2 in Nature Genetics, 8 in the American Journal of Human Genetics, 4 in other journals. 5 more manuscripts are currently under review and 17 are in preparation. We think this has been quite successful. We think it's been successful because it values clinicians, it exploits existing strengths in both clinical genetics and genomics, but neither group exploits the other. It really is a collaboration. The work and the resources have been shared nationally. Patients from all over the country have been studied. It builds on prior collaborations and trust among the participants. It's had strong leadership. Kim Boycott has done a terrific job in that regard, but shared decision-making, and there have been effective cooperation among the funding agencies. And this is actually a key issue. This is a unique example where, because actually CIHR put in the biggest part to start with, Genome Canada didn't require any other matching besides CIHRs. We never could have done this so quickly if we had to assemble matching from multiple places. So this collaboration between CIHR and Genome Canada was terrific. Genome BC, Ontario Genomics Institute, McLaughlin Institute, Genome Quebec all have come to the table and enlarged the pool and enabled us to do much more. So we're very pleased with the progress we've had.